Welcome to the deep dive. Today, we're taking the ultimate intellectual shortcut into a universe you can't even see.
A completely invisible universe.
The fundamental unit of life itself. We've stacked up your sources, research papers, diagrams, all the historical context, and our mission is to map out this microscopic machine, the cell.
And to really get how it performs the miracle of life.
It really does feel like a miracle.
It is. I mean, if you want to be well-informed about biology or medicine or even just how your own body works, you have to start here.
It all comes back to the cell.
Always. It's the basic structural and functional unit of every living thing. It's the smallest entity that can do it all. Manage energy, get nutrition, remove waste.
And reproduce.
And reproduce.
Yeah.
Everything.
And the really wild thing is just how long this basic reality was, well, hidden from us. Our tour begins not with some high-tech lab, but with a piece of cork.
Back in 1665.
Way back.
That's right. That was Robert Hooke. And he wasn't a professional biologist, not really. But he was this incredible inventor.
He built his own microscope, right?
A crude one. Yeah, a self-designed compound microscope. And he used it to look at a super thin slice of cork.
And what he saw changed everything.
He saw a grid. Just a grid of tiny, empty, box-like structures.
The famous honeycomb pattern.
Exactly. And he thought they looked like the small rooms that monks lived in. So he coined the term cells, from the Latin word cella, which just means a little room.
That's amazing. And he was actually looking at dead cell walls.
He was, yeah. He was seeing the leftover structure, the skeleton of the cell, you could say.
So the first glimpse was of an empty room. Seeing actual living, moving life came almost a decade later.
That's when Anton von Leeuwenhoek enters the scene, 1674. He had a much, much better microscope.
Just a single lens, though.
A single lens system, but it was remarkably powerful for its time. And he was the very first human to see free-living cells.
What does he call them?
He called them animalcules, these little animals swimming around in pond water in his saliva.
Even scraped from his teeth.
Right. He saw life in motion for the very first time. It must have been just mind-blowing.
So the knowledge builds slowly after that, right? You have these initial sparks, but then the scientific world kind of takes a breath.
A very long breath. It took over 150 years before the concept really crystallized into a proper theory.
In the late 1830s.
That's when the official cell theory was formulated. You had Matthias Schleiden in 1838 saying, all plants are made of cells.
And then Theodor Schwann followed up.
In 1839, confirming that yes, all animals are too, and they defined the cell as the basic unit of life.
But there was still a missing piece, a huge one. Where do cells come from?
That final critical piece came from Rudolf Virchow in 1855.
With the famous postulate.
The famous postulate, omnis cellula e cellula.
Which means?
All cells arise from pre-existing cells. That established the continuity of life. And the three-part theory is, well, it's the bedrock of all modern biology.
Okay, so let's unpack that bedrock. Let's start with the outer defenses. Every single cell has to define itself, and that starts with the plasma membrane.
The cell membrane.
We can think of this as the cell's, I guess, its sophisticated border control.
That's a great analogy. It's perfect, because it's not a passive wall. It's the outermost covering that maintains homeostasis.
Internal stability.
By separating what's inside the cell from the world outside. And its most important feature, as you noted in the sources, is that it's a selectively permeable membrane.
Meaning it doesn't just let anything pass, it chooses.
It chooses what comes in, what goes out, and that power comes from its structure.
Which is made of what?
It's primarily made of organic molecules, lipids and proteins. The lipids form this double layer, like a fluid sea.
And the proteins are floating in it.
The proteins are embedded within it, yeah. Sometimes they span the entire width. They're like gates or sensors.
I remember seeing diagrams of this, the fluid mosaic model. Does that still hold up?
Absolutely. It's dynamic, it's flexible, it's constantly moving, and that flexibility is so important.
It's not just about shape.
No, it lets the cell do amazing things. It enables endocytosis, where the membrane literally folds in on itself to engulf something big like food.
That's how an amoeba eats.
Exactly how an amoeba eats.
Okay, so that's for taking in the big stuff. But what about small molecules, like gases? The most basic transport has to be diffusion.
Diffusion is the simplest.
Yeah.
It's just substances moving from where they're highly concentrated to where they're less concentrated.
Passive transport. No energy needed.
Zero energy.
Yes.
It's like a ball rolling downhill. It just happens.
And it's absolutely vital for survival.
Well, think about breathing. A cell's waste, like carbon dioxide, builds up inside. The concentration gets high.
So it just naturally flows out.
It diffuses out into the lower concentration environment. And the reverse happens with oxygen. The cell is always using up oxygen, so its internal concentration is low.
So oxygen just flows in?
Spontaneously diffuses right in to fuel the cell.
Okay, so that handles gases. But what about the most important molecule of all, water?
It follows the same rule,
Exactly.
but we give it a special name when it crosses that selective border: osmosis.
Right.
Osmosis is the net diffusion of water across a selectively permeable membrane toward a region of higher solute concentration.
So water always tries to dilute the side that has more stuff dissolved in it.
That's the key. Water moves to dilute.
Okay, this is where the concentration game gets really high stakes for a cell. Understanding these three outcomes is it's crucial.
It is absolutely the difference between life and death. So let's start with a hypotonic solution.
Okay, what does that word?
It means the environment is very dilute. The medium outside the cell has a higher concentration of water than the cells inside.
So water rushes in to try and dilute the cell's guts.
Precisely. Water floods into the cell, causing it to swell up. This is why plant roots are so good at absorbing rainwater.
But for an animal cell, that could be bad.
Very bad. Without a rigid wall, it can swell until it bursts. It's called lysis.
Oof. Okay, what about the opposite of that when things are in balance?
That's an isotonic solution. The water concentration is equal on both sides of the membrane.
So nothing moves.
No, water molecules are still moving back and forth, but there's no net movement. The cell stays the same size. This is why hospital IV drips are isotonic with your blood.
They don't want your blood cells to swell up or shrink.
Exactly.
Which brings us to the final worst-case scenario, the hypertonic solution.
Right. So in a hypertonic solution, the outside world is very concentrated. Lots of salt, lots of sugar, which means
It has a lower concentration of water.
a lower concentration of water than the cell's interior. So the cell now has the higher water concentration.
The water moves from high to low.
So water rushes out of the cell and it shrinks. It shrivels. This is why drinking seawater is so dangerous. It literally pulls the water out of your cells.
That really clarifies how much work that membrane is doing. So the membrane is the border patrol, but some cells need like heavy-duty armor.
And that's where we get the cell wall.
Right. This is what really distinguishes plants, fungi, bacteria.
It's a rigid outer covering that lies outside the plasma membrane. It's what gives a plant its actual structure.
And in plants, that rigidity comes from cellulose.
An incredibly strong, complex carbohydrate. Its main job is structural support, but it also creates a unique way to deal with the environment.
What happens when a plant cell is in that dangerous hypertonic solution?
It still loses water, but the whole cell doesn't shrivel. Instead, the cell contents, the plasma membrane, and everything inside just pull away from that rigid wall.
That's a specific process, isn't it? Plasmolysis.
That's plasmolysis.
Yeah.
But here's the key protective part. Remember how animal cells burst in a hypotonic solution?
Right, they pop.
Plant cells don't. When water rushes in, the cell swells, but the rigid wall pushes back with an equal and opposite pressure. It can't burst.
It's a safety harness.
A perfect safety harness. It allows the plant to thrive even when it's flooded.
Okay, speaking of fundamental differences, this is the perfect jumping off point. The two great systems of life. Prokaryotes versus eukaryotes.
This contrast defines, pretty much the entire evolutionary trait.
Let's start with the simple ones, prokaryotes.
The name means primitive nucleus, and they are tiny, usually 1 to 10 micrometers. Their defining feature is a lack of internal compartments.
No true nucleus.
Correct. They have a region called the nucleoid, where their DNA just kind of floats around. And critically, they have no membrane-bound organelles.
They're minimalists.
The ultimate minimalist. Bacteria are the classic example.
So what about us, the eukaryotes, the true nucleus cells?
Eukaryotic cells are giants in comparison. 5 to 100 micrometers. They are defined by their internal structure.
They have a real nucleus with a membrane.
And they have dozens of specialized membrane-bound organelles that create a division of labor. Fungi, plants, animals. We're all in this category.
So let's take the tour inside one of these complex eukaryotic cells. We have to start at the command hub, the central office, the nucleus.
The nucleus is where all the blueprints are kept.
Yep.
It's surrounded by a double-layered nuclear membrane, and it has pores to control what comes in and out,
And inside are the chromosomes.
which are made of DNA and protein. And the functional segments of that DNA, the instructions, are called genes.
So the nucleus is the CEO.
It is. It directs all the chemical activities. It controls growth, reproduction. If you mess with the nucleus, the whole operation falls apart.
Every city needs power, and in the cell, the powerhouses are the mitochondria,
They're the energy converters. They generate usable power for everything the cell does.
in the form of ATP?
Adenosine triphosphate, ATP.
Hmm.
We call it the energy currency of the cell. If the nucleus is the CEO, mitochondria are the treasury, printing the money for every single job.
You noted in the sources that mitochondria have a secret. They have their own DNA and ribosomes separate from the nucleus? Why?
This is one of the most incredible ideas in biology, the endosymbiotic theory.
Okay.
It suggests that billions of years ago, mitochondria were actually independent bacteria. They were engulfed by a larger cell, but instead of being digested,
They stuck around.
they formed a partnership, a symbiosis.
Yeah.
And they kept their own tiny circular genome, which is why they're semi-autonomous.
So our power plants were once invaders who decided to stay and help.
It's an amazing story. Now let's move from power to the manufacturing and shipping network, starting with the endoplasmic reticulum, or ER.
That sounds like a massive highway system.
It is a vast interconnected network of tubes and sheets, and there are two types.
The first being the rough ER.
It looks rough because it's studded with these little black dots.
Ribosomes.
Ribosomes, and that's no accident. The RER is the primary site of protein synthesis, especially for proteins that are going to be shipped out of the cell.
And the second type is the smooth ER.
The SER has no ribosomes. Its jobs are more specialized. It manufactures fats and lipids which are needed for membranes.
And it has another really important job.
A crucial one. The SER plays a huge role in detoxifying poisons and drugs. This is why your liver cells are absolutely packed with smooth ER.
So the ER is building the parts, and the actual factory workers are the ribosomes.
The ribosomes are the ultimate protein factories. They exist in all cells, prokaryotes, eukaryotes, they're everywhere. They're the machines that read the genetic code and build the protein chains.
Once those proteins and lipids are built, they need to be processed and shipped.
And that job belongs to the Golgi apparatus, the cell's post office.
Okay.
It's a stack of flattened sacks called cisterns. Material from the ER arrives at the Golgi, and its functions are storage, modification, and packaging.
So it's like an Amazon distribution center.
Exactly. It takes a raw protein, maybe adds a sugar to it, and then packages it into a little vesicle for dispatch.
And some of those packages become the cell's waste disposal crew, the lysosomes.
They are membrane-bound sacs filled with incredibly powerful digestive enzymes.
Made by the RER.
Made by the RER, packaged by the Golgi. They digest foreign material like bacteria or just old, worn-out organelles that need to be recycled.
They're famously called the suicide bags of the cell. Wait, the cleanup crew has a self-destruct button? Why?
It's a necessary fail-safe. It's for controlled cell death or apoptosis.
Program death.
Right. If a cell is badly damaged or infected, it's safer for the whole organism if that cell just removes itself cleanly. The lysosomes can burst and digest the cell from the inside out.
Wow. Okay, before we finish our tour of the city, we have to look at the structures unique to plants. Let's start with plastids.
Plastids are essential for plants. And again, just like mitochondria, they have their own DNA and ribosomes, a huge evolutionary clue.
And we can group them by color.
We can. You have leucoplasts, which are colorless and mainly for stored starch, oils, proteins.
And then the most important ones, the green ones, chloroplasts.
Chloroplasts contain chlorophyll, the pigment that captures light energy. They are the sites of photosynthesis.
They're the food factories for the entire planet.
Absolutely.
And finally, plants have these massive storage tanks, the vacuoles.
Huge vacuoles. While an animal cell might have small ones, in a mature plant cell, the central vacuole can take up 50 to 90% of the cell's volume.
What's in it?
It's filled with cell sap, a watery solution of sugars, amino acids, waste. But crucially, the pressure of this vacuole pushing against the cell wall is what gives the plant its rigidity.
So when a plant wilts, it's because the vacuole has lost water.
That's often exactly what's happening.
We've seen how cells are built and how they run, but if they didn't reproduce, life would end. So how do organisms grow and repair themselves? Through cell division.
And there are two distinct methods depending on the goal. The first and most common is mitosis.
For growth and repair.
Exactly. This is how you heal a cut or how a baby grows. The mother cell divides into two identical daughter cells with the exact same number of chromosomes.
A perfect copy.
A perfect copy.
But the second process is totally different. It's all about sexual reproduction.
That's meiosis. This only happens in the reproductive organs to form gamete, sperm, and eggs. It involves two divisions, one after the other.
And the end result is different.
Very different. It produces 4 new cells that have only half the number of chromosomes as the original cell.
Why half?
Because when the sperm and egg combine during fertilization, the chromosome number is restored to the full count for the offspring. It's a brilliant system.
So we've toured this miniature world, and the takeaway is just, it's profound. The cell's incredible efficiency comes from the coordinated organization of its membrane and all these specialized organelles.
And if you connect that organization to the bigger picture of your own health, you start to see why this is the key to so much of modern medicine.
Because when these microscopic parts fail,
The consequences for the whole organism are immense.
give us an example. How can one tiny factory failing derail the whole system?
Okay, think about the mitochondria, the powerhouses. There are dozens of recognized mitochondrial disorders, all caused by tiny errors in their DNA or function.
And because they provide the energy?
Because they provide the ATP currency for everything, these disorders can show up as almost anything. Muscle weakness, neurological damage. The most energy-hungry organs suffer first.
It's just incredible to think that a failure in a power plant, one that might have once been a free living bacterium, can cause system-wide failure in a human.
Absolutely. And the same goes for the production line. There are ribosomopathies caused by ribosome malfunction that can lead to developmental defects
Or cancers.
or certain cancers, and problems with the detoxifying smooth ER in your liver cells are directly linked to chronic conditions.
Oh.
Understanding this basic miniature organization, how traffic flows, how power is made, how waste is managed, is foundational
It's the key to preventative medicine.
and to unlocking future treatments.
So stay curious about cellular pathology, because that organized chaos we just explored is truly where the future of medicine so often begins.